I had a question.
Sure, go ahead.
That's as simple as you want us to go towards the anterograde and the retrograde transport, right? With the dynein and the kinesin motor.
Yep, that's it.
Okay. Thank you.
You're welcome. All right. So now we have our glial cells. And the glial cells, obviously, as we briefly mentioned earlier, as you remember from anatomy, they're there to support. Okay. They protect, they help maintain these nerves and allow them to do what they do. We have glial cells that are found only within the peripheral nervous system. They're on this slide. Schwann. And satellite cells. We have ones only found in the central nervous system. There's 4 of those. Okay. You should know either one of those terms there for Schwann cell. Okay. You should know it as a Schwann cell, which everybody on planet Earth generally knows it as, but it also has a very fancy pants name called a neurolemmacyte. And you should know that one as well. Okay. They are used interchangeably between textbooks. Even I think between some textbooks, they'll, even switch back and forth between that term. Okay. So be comfortable with using both. For satellite cell, just call it a satellite cell. Okay. You can see it has another name, ganglionic gliocyte. Don't worry about that one. Okay. Just call it a satellite cell. Schwann cells or neurolemmacytes, they're the myelin producing cell. Okay. They're also going to form that neurolemma as well. Okay, that outer connective tissue covering surrounding an axon. So, spoiler alert, they also do that. Think of the Schwann cell like a sock in that regard. Satellite cells, all they do is kind of surround that ganglion, all of those nerve cell bodies out in the periphery there. And they're obviously there to just kind of help support and protect that little glial cell. We're not going to spend our time talking about it. We're going to be talking a lot about neurolemmacytes. That's going to be really important for you to remember what the world that guy does. When it comes to, excuse me, when it comes to the central nervous system, we have a whole menagerie of neuroglial cells. Oligodendrocytes, microglia, astrocytes, ependymal cells. All of these are vitally important for what the nervous system is allowed to do. Oligodendrocytes, they're the myelinators. Okay. They are the myelinators. When it comes to oligodendrocytes, when it comes to oligodendrocytes can myelinate multiple nerves, multiple axons. Schwann cells are only found along one stretch of an axon. Okay. So in the periphery, it takes a lot of Schwann cells to myelinate a axon. As you can see on the oligodendrocyte, it can myelinate many. It gives off these cytoplasmic extensions, and there you can see it wrapping that fat around that axon. Microglia, you really only see microglia elevated in a spinal tap if you have an infection within the central nervous system. So these are your immune cells of the CNS. Okay. So they're help to get rid of any foreign debris. If there's any damage, you'll find microglia involved in that as well. But generally, we think of it in terms of an immune type of cell. So if you take a spinal tap and you find a lot of microglia in it, you have an infection of the central nervous system. Astrocytes, these are the dudes that help form that blood-brain barrier. Okay, but they also do a number of other things as well. And I think I have maybe some listing about them in a little bit, but they help to regulate entrance of materials from the general circulation into the circulation to the central nervous system. They're involved with guiding neurons during development in utero, make sure the neuron gets to the right spot. I think they have some involvement with synaptic formation, ensuring that it happens properly. So astrocytes do a lot of heavy lifting. Generally, we only talk about it in the term of the blood-brain barrier, though. And I think I have some stuff sprinkled in a little bit later. Last one is ependymal cells, and ependymal cells line the spaces within the CNS, the ventricles. And they do one thing, they secrete CSF, cerebrospinal fluid. Okay. How many ventricles do we have? Anybody?
Like in the heart?
Huh?
Like in the heart?
No, within your brain.
Oh. Two
Lateral ventricles, right? Left and right.
3.
Third ventricle's in the midbrain. 4. Fourth ventricle is there between the medulla oblongata and that space in between it and the cerebellum. So we have 4 different ventricles there within the central nervous system within the brain. And then that fourth ventricle continues down through the spinal cord as the central canal. Gemma was just getting ready to log in to say that. I saw that. I see you. So those are our 4 dudes. Okay. There's the ependymal cells lining the ventricles, lining those spaces, producing CSF. There's the CSF that bathes spinal cord in the brain, buoyancy, provides nourishment, all that fantastic stuff. There's the astrocyte. Okay, looks like a star, hence the name. Has these little feet-like things, perivascular feet, they call them. Helps form an even tighter barrier around those blood capillaries. Here's your microglia, looking for dudes to kill. And there's a table with all of it listed down for you in case you don't like flipping back and forth between slides. But myelin is a pretty important thing here. Okay? Myelin is a pretty important thing here. So understanding a little bit about the neurolemma and that layer of fat is pretty important. And if you kind of unwrap that Schwann cell, there's what that big flap of a structure looks like. Okay? The degree to which this thing extends, cytoplasmic extensions wrapping around this thing, gives us our myelinated appearance to some nerves. And if you look on an imaging study here, you can see, okay, there's all of that's myelin. And then, you know, if you parse it out, and really, you could see multiple layers of fat. Okay? That really helps to insulate this thing. And insulation helps to speed up nerve impulse conduction. Helps to speed up electricity, right? You've ever seen the wires in your house? They're coated in plastic. Okay? Helps to speed up electricity conduction within the house. If there's an open spot on that plastic and you touch it, magic, right? Electricity flows with you. Down here, you can see an unmyelinated axon. Still has a Schwann cell around it. Okay, still has a Schwann cell around it. It's just not layered. Okay, it doesn't continue to wrap and wrap and wrap. It doesn't continue to grow and grow and grow, wrapping itself around the nerve, wrapping it around the axon. Okay. So all axons are surrounded by these Schwann cells. Okay, neurolemma, I believe, kind of, I think, translated out to a sheath of Schwann. Okay. So all of them are going to have it. You know, even unmyelinated axons. Okay, they'll have a Schwann cell wrapped around it, but it's only wrapped around it once, and that is what creates that neurolemma. That's what creates that neurolemma. It's when it continues to grow and wrap around this axon, this stretch of axon, where it gives it all of those fat layers that gives it that true myelinated axon that you come to think of when you think of what a Schwann cell does. As you can kind of see there, I'm not going to ask you, but there's the size. Very small axons are generally unmyelinated. It's your bigger ones, your bigger nerves, bigger axons, medium-sized axons that are going to be the ones more associated with Schwann cells or neurolemma and myelin at the same time. And obviously the important aspect about myelin is that it helps to speed up that nerve impulse conduction. I guess I should put here. Come back. Okay. And again, it does take many of these to myelinate axon on that periphery, which again is very different from an oligodendrocyte. Okay. An oligodendrocyte can myelinate many different axons, right? That is a significant difference between the two. Okay. One oligo can wrap several axons. Okay. And when we look at the central nervous system, we see areas of white, see areas of gray. The gray matter is unmyelinated portions of the axons clumped together. These are generally nerve cell bodies. These are generally unmyelinated axons, but a lot of nerve cell bodies in these regions. And when we look at the brain, right, that gray region or that gray matter region is on the outside. Okay, it makes up the cortex. That's where all the nerve cell bodies are. And then we have smatterings of gray within the middle part. Those are your nerve cell bodies there. Putamen, globus pallidus, specific nerves that ensure that information gets to the right location. In the spinal cord, the gray matter is centrally located, okay? And the white matter is on the periphery, so it's backwards. Okay, it is backwards. And obviously white matter has myelinated axons. So a lot of nerve tracts in those regions. There may be some clinical pearls that I ask on the exam. If that is the case, I will say this is something that you need to know for the exam. This is not necessarily one of them, though. If I was going to ask it, it would be for bonus. So don't spend 5 days trying to memorize this slide, please. But you can have demyelinating conditions, diseases. Multiple sclerosis is probably the most common one that we have there. And this one's an autoimmune disorder. Okay, it's autoimmune, so the body's going to attack itself. All right, so it targets that myelin sheath and destroys it, causes sclerotic damage, and that can affect how nerve impulse travels down through that length of axon. Okay, depending on the degree of sclerotic change, depends on if an impulse action potential gets through it all. And you can have waxing and waning of, how the body is attacking and the degree to which you have a flare-up of MS. It happens, it affects women more than men as well. Usually kind of manifest in the 20s, early 20s, is kind of where it generally starts to manifest, 30s maybe as well, but generally in the early to mid 20s. Guillain-Barré syndrome is, again, the body's attacking myelin sheath. Very rapid onset. This causes some pretty significant muscle weakness as well, but there are a couple different ones there. MS, there may be some genetic links to it as well. So some interesting things involving those myelinating cells. Mercury poisoning does the same thing, accumulates in the fat around those axons as well, and the body tries to clean it up and it destroys the length of axon, damages it pretty severely there. So that can affect growth and development, obviously. That's why your teacher in high school was always like, if you drop the thermometer, they're always like, back away. They didn't want you even touching it, because as soon as you touch it, you absorb it into your skin and it gets into your central nervous system. That's not a good thing. What happens if you cut? You can cut a nerve, you can damage a nerve, right? There are a number of proteins, number of factors, if you will, that are going to be associated with this process. There's the no-go one that's produced by oligodendrocytes. You have nerve growth factor. You have brain-derived neurotrophic growth factors, glial-derived neurotrophic factors, neurotrophin 3, neurotrophin 4, neurotrophin 5. There's a lot there. I'm really only asking you what's on this slide. Okay, the book goes into a very huge amount of detail looking at this. They look at some potential studies, why it's a little bit easier in children to do it as compared to adults. I just want you to know who's involved. Who's involved? The Schwann cell. Okay. The Schwann cell is going to release some various different types of growth factors that's going to allow this process to occur. In research, in regeneration of nerves, regrowth of damaged nerves, we have a lot of success, like a ton of success. I remember back in the 90s, before you people were probably born, there was this big study that came out. Okay, they took these rats, took a scalpel and cut their spinal cord. May comprise some biological cement, glued the two cut ends back together, and within some pretty short time, these rats that were couldn't walk in their lower legs were able to grow connections back within their spinal cord. And that was like one of the first kind of major studies that looked at it. Awesome. Everyone's like, we got it. We figured it out. Biological glue, stick the two ends back together, and magic, they can grow back together. Is that a normal mechanism of injury, though? Being cut by a scalpel? I mean, there's sociopaths out there, right? Sam, do you think it's common?
To be like cut by a scalpel?
Yeah.
I mean,
yeah, like if you trip and fall down some stairs, or you dive into the pool.
No, there's not going to be like a rogue scalpel, but people do have a lot of surgeries, just like in general. So I would figure,
they do.
Yes.
And I'm going to pray to gods, all of them, that doctor doesn't cut my nerves. Okay, so when you damage. When you damage something, you jam it, and there's a lot of twisting and shearing. So, there's never a cut that's just a nice little cut. I can just glue those right back together. There's twisting, there's fraying, and then various different structures there in the CNS or the PNS, respectively, are going to go and clean up all that debris. And now I have a huge distance between those two. Well, you're not built with slack, okay? So I can't just also, bring the slack together and biological cement and regrow. So it takes a long a substantial long period of time to grow this little, what do they refer to it as? This little sprout, okay? This little axon sprout. It takes time to regrow this. And this is an artist rendition of if everything goes to plan. And it can reestablish this. I am not saying that it can't, but the degree to which this site of injury is from the nerve cell body is significant. The farther away, the longer it takes that material to go in that anterior grade direction, right? The longer it's going to take to send all the waste, debris that it's cleaning up inside of itself. Okay. So it takes time to get this little sprout to emerge out. And it only grows like 1/16 of an inch in a year's time. So, I mean, this takes some pretty significant time to regrow. And when you do eventually make a connection, hopefully, it's usually never the same. Okay, the axon, the dendrites that emerge off of it are generally thinner and weaker. And when I mean weaker, it just can't conduct a nerve impulse as effectively as it once could. It makes sense, right? I mean, it's fibrotic damage that occurs with any type of injury. The Schwann cells, okay, that regrow are much more smaller. They're not as robust. Okay. And obviously the amount of myelination that those things can have can affect how a nerve impulse goes. So there are a number of things that go into this, Wallerian regeneration type of process here, right? But if you're going to have success, it's going to be successful out in the periphery. In the central nervous system, again, we have blood-brain barrier. We have astrocytes. We have microglia. There's going to be a lot of swelling. There's going to be a lot of damage. And then we're not going to be able to as effectively get all those machinery, those parts and pieces, the raw materials out into that, through that blood-brain barrier and into the brain or into the spinal cord. Okay. Also, too, there are a huge number of growth factors that you see in utero during fetal development that you do not get anymore. Okay. Once the brain grows during that time, and once mom spits you out, cuts the cord, you don't have all of those growth factors that you saw in utero at certain specific times. And so the ability to regrow this quickly and effectively, bye-bye. So if you do have some severe damage to axons within the brain, hopefully, I don't want you to have it at all, but if you're going to have it, having it done younger, before wires are hardwired in the brain, for this track only goes here, okay, as opposed to having damage done to the brain, axons, as you're an older person. Those lines of communication are pretty hardwired at that point. So it takes time to relearn those type of things. Okay? Walking, talking, moving effectively. All of that takes a hell of a lot of time if it's a spinal cord injury itself as compared to a spinal nerve injury. So when it comes down to it, guys, this is what I'm asking. Even the book is going to go into a number of details about it. There are receptors on these axons as well that can promote apoptosis of an oligodendrocyte. So it can promote death of even more oligos as well, as it's trying to repair itself. So again, there's a whole host of things that goes on. Who's involved? Schwann cells. Okay. Who's involved? Schwann cells. Why is it so difficult? Why is it so difficult in the central nervous system compared to the peripheral nervous system? Because of that BBB. because I don't have access to all those stimulating growth factors that you see in utero as the brain is developing. That's why CNS is such a hard nut to crack. Here's the astrocyte, and I believe I have some of the functions again on that next slide. But there it is. You can see you always find astrocytes in close proximity to a nerve and in close proximity to capillaries. Okay. They help to kind of hold and govern the interaction between blood and the nerves. Okay. It's them that help to create an even tighter seal between that little capillary. Because remember, we have three types of capillaries, right? We have capillaries that have a continuous sheet where we have no spaces. We have fenestrated. We have sinusoidal capillaries. Okay. And with this type of capillary in the brain, it's more of the continuous type, right? We have some nice tight junctions between those little endothelial cells. Oh, fantastic. But then we have these perivascular feet extending off of that astrocyte that help to make that influence even tighter, right? And really help to prevent everything in the general circulation getting in and affecting the brain chemistry. Because there's a lot of stuff that can affect the brain chemistry, and I really want a nice controlled environment. Okay. And you could see it even does some nonsense with kind of reabsorbing some specific neurotransmitters, glutamate. Okay. And we'll talk about glutamate, definitely not today. We'll talk about that Thursday. But here are some of these fantastic functions. Where is it? Right there. We keep talking about, glucose as the preferred energy source for the body, specifically red blood cells in the brain. What happens if you go, no carb or low carb? Well, it's these guys, these astrocytes. They're involved with helping to create this interaction, getting that lactate, getting it into the neurons, to the central nervous system for them to use. So they're the individuals involved with that process. And that takes a little bit of time. That's why one of the most common side effects of keto, low carb, is that brain fog, because it takes time for these astrocytes to allow that phenomenon to occur and for them to kind of exhaust their little stores of it and then start to make new, because that takes time, right? And that's where these two kind of go hand in hand. As you can kind of see, it's involved with helping to re-up the reuptake of some neurotransmitters, specifically glutamate. So it's going to help with some inhibitory functions associated within the brain. And we'll look at that when we look at glutamate in the next lecture period. Again, it helps to maintain a nice, stable environment. Okay. It helps to create that situation there to maintain those ionic ratios. Sodium is great on the outside, potassium is great on the inside. We have a lot of potassium floating around. Okay. And it's astrocytes' job to help maintain the status quo there of keeping sodium the greatest cation that we have there and not potassium. I made reference to it earlier. It helps to ensure that nerves get to the right locale during fetal development. So it's involved with brain development very intimately. And you all know the last one. Okay. That's the one that you probably definitely learned in anatomy class. And that's going to make that blood-brain barrier. What in the heck is the blood-brain barrier? Okay, well, there it is. Okay. Nice tight junction, super tight junction between the capillary and the structures of the central nervous system. Okay. Very selective process. Only certain things can move across. Okay. We can move some other things with some other various mechanisms. But again, it's going to be very selective. Okay. Very selective. That's why, treating, brain cancers is very difficult, okay? Because those astrocytes limit a lot of that movement across. Astrocytes are very involving the breakdown of toxic substances within the blood. Okay, so that's why treating brain cancer is such a very difficult, arduous process to do.